Hello students, I am again back with another part of my video on the topic heredity and evolution. This is the fifth video that I am about to upload. In fact I have uploaded, right? Okay. So now today in this video I will be talking about monohybrid cross. Okay. Monohybrid cross, it was a part of experiment done by Mendel. Now, what exactly is monohybrid cross? Okay, it is in fact a cross done between two organisms of a particular species, keeping in view or to study the inheritance of a pair, a single pair of traits for a particular character. Okay, so what exactly is monohybrid cross? For example, I'll just tell you. For example, I'm going to take one character. I'll keep in mind one character, that is height. Okay. Now this height of the plant or any organism can have two traits. It can be tall, it can be short. Okay. So now I have to study the inheritance of these two traits or these two alleles. Okay, a single pair of traits or two contrasting traits for a particular character. So whenever a cross between two organisms of a species is done to study the inheritance of two contrasting traits or a pair of contrasting traits for a particular character is known as monohybrid cross. Okay, so I'm just writing the definition. All of you please note it down. Monohybrid cross. Monohybrid cross. A cross done between two organisms of a species to study or to observe the inheritance of a pair of traits, a pair of contrasting traits, or you can even say two traits, two contrasting traits of a character. Okay, now this is what we call as monohybrid cross. Okay, so this was the very first experiment that Mendel also did. Now what did Mendel do exactly? Mendel took pea plants and he basically took into consideration one character, that was the height of the plant. Okay, and he took two categories of plants, tall plants and short plants, right? There were two types of plants that he got, tall and short plants. So what did he do exactly? I'll tell you. Initially he took some tall pea plants. I'm just going to make this something like this. Okay, so he took these tall pea plants and he did self pollination, okay, of these pea plants only, and he in fact crossed these plants with each other and only the tall plants. Now after crossing, in the next generation he got some plants which were tall, okay, some plants which were tall, and there were some short plants also in between, right? There were some short plants in between. So what did he do? He removed these short plants. Okay, he removed these short plants, he just segregated it, and then he took again these tall plants, and again he self-crossed these tall plants. Again he got, tall plants, tall, but there were few short plants also in between, right? Very few short plants. So the number of short plants kept on reducing, and this way, he kept on doing this until he got all tall plants. Okay, so what did he do? He took all those tall plants. Now, why did he do that? Why did he, he kept on crossing the tall plants or self-crossing the tall plants just to get pure tall character. Okay, he wanted that the tall plants that he is taking, it should have the factors which promote only tallness. Okay, he did not want any plant having factors which would promote shortness as well, right? So he took all the tall plants, and you call it as pure tall. Okay, pure tall, right? So he took pure tall plants, and he also took pure short plants. The same work was done with short plants also. He took those short plants, and then he self-pollinated the short plants, and then he got some tall, some short plants. He segregated the tall plants. Again, he took the short plants. Again, he did self-crossing between the short plants, and he kept on doing this till he got all short plants. Okay, so we can say that he took pure tall and pure short plants. Now, I think it's very clear to you why I am calling it as pure tall and pure short. Pure tall means that all the alleles which promote tallness, or all the alleles which basically is related to height, is of tallness. Okay, so that's why I am calling it as pure tall. Okay, and the other word for pure is homozygous, right? So this you have already gone through in my previous videos, right? So he took pure tall and pure short plants, and he named them as parental generation. Okay, parental generation. So see what happens whenever you have to study the inheritance. It has to start from somewhere. Okay, so he started from parental generation. So wherever we start from, that particular generation is known as the parental generation. And what is so special about the parental generation? That parental generation have pure traits or pure character. Okay, they are pure, right? So we have pure tall and pure short plants. Now he crossed the pure tall with pure short plants. So when I'm saying this word cross, now what exactly do you mean by cross? Cross is nothing but cross pollination. Okay, so what I'm talking about is cross pollination, right? So he crossed tall plants with short plants, and then he collected all the seeds, and in the next generation the type of plant that he got were all tall plants, and the next generation is known as the first filial generation. Okay, you can also call it as F1 generation. Okay, you call it as F1 generation also. So the first filial generation or the F1 generation, all the plants that he got were tall plants, right? Now definitely this was very surprising for Mendel, because there were short plants also which he used in this experiment, but nowhere he could find the trait of shortness in the first generation. And he was wondering where this particular trait went, or what happened to this particular trait. Okay, well, now he continued this experiment. So but before we move on to this particular experiment as to what happened and how did this happen, see, actually what happened is, if we do the genotypic crossing, if we show the crossing genotypically, or the genotype of this all the traits, and then we do the crossing, we will understand as to how this particular trait came into existence. But with this experiment, what did Mendel conclude? Mendel concluded that whenever there is a mixture or there is a combination of two different alleles for a particular character, one of the allele shows that its trait, or express its trait or express its character, and that allele is known as a dominant allele, and the other one, which is not able to express itself, is known as the recessive allele. Okay, so we come to two terms here, that is a dominant trait or a dominant allele, or a recessive trait or recessive allele. Okay, so what is a dominant trait and what is a recessive trait? Dominant trait is a trait which is always expressed in the presence of a recessive trait. So in a heterozygous condition, rather, always a trait which is expressed is known as a dominant trait, and the trait which is not expressed is known as a recessive trait. Okay, so a recessive allele will never able to express itself. So when I am talking about expressing, what do you mean by expressing? Expressing means that the character is shown physically. Okay, so suppose if you have a gene of tallness and a gene of shortness in your body, but you are tall, so that means the gene of tallness has expressed itself phenotypically, and that's why you are tall, and the gene of shortness could not express itself phenotypically or physically, hence you are not short. Okay, so you are tall because of that, right? So this is what the law of dominance is. Now usually this law of dominance, there is nothing like law of dominance in CBSE. In your ICSE you have this law of dominance. Okay, in CBSE we have the law of segregation under monohybrid cross. Okay, but here definitely you should know about the dominant trait and the recessive trait. Okay, now before I move ahead, I would like to discuss something with you people. You remember when in my previous videos, when I was just talking about the traits of when I was just talking about homozygous heterozygous condition, and I was giving you an example of Amitabh and Jaya, and I made the alleles something like this. Okay, tallness and shortness and all these things. That was just to show you, that these are the alleles, but you cannot show alleles with these lines every time. It's quite impossible. Okay, so initially maybe in the previous videos I made these particular lines to show you the alleles, but it should not happen like this. So the thing is, how do we express the genotype of any particular character or any particular trait? Now, to express the genotype of any character or any trait, we need to write the alleles. We need to write the alleles. Okay, and how are these alleles written? These alleles are actually written, or they are basically shown by English alphabets. Okay, they are shown by English alphabets. Now, how do we show these alleles with the help of English alphabets? See, there are several ways by which you can do. Like for example, like in IGCSE, I am also teaching the IGCSE board. There usually a trait is, for example, height is a particular character. Okay, height is a character. So and it has got two traits. One is tall and other is short. Okay, so you can express it like this: H and T and H and S, if you can express it like this, or you can express it H capital T, that would depict the tallness, and H small t, that would depict the shortness. So short can be depicted like this, tall can be depicted like like this, okay, or you can even write S over here. So this is a way of depicting or showing the allele, right? But in CBSE and especially in grade 10, when you are going to show the crossing, how you are going to express the genotype. Now, to express the genotype, you should first know about the dominant and the recessive trait. Okay, so you should know which trait is dominant and which trait is recessive. Okay, so here, for example, when I am taking height, I know that tallness is the dominant trait and shortness is a recessive trait. Okay, now to depict height, I am going to take the first letter of the dominant trait. Okay, I will be taking the first letter of the dominant trait, and what is it? T. So the alphabet that I am going to use to depict this particular character will be T. Is it clear? It will be T. Now, for a dog, now for any trait, always remember I told you that there are two alleles which are responsible to give you a particular trait. So when you are expressing dominant, you are going to write it this way. That is, these are the two alleles to depict dominant trait, that is capital T and capital T. Okay. Now, when it comes to recessive trait, since it's the same character, shortness is a trait for height only, and we decided that we are going to depict this particular character with letter P. So here, to depict a recessive trait, I am going to use these alleles. So it means that a recessive trait is expressed by small t and a dominant allele is expressed by capital T. Okay, and and why do we use the same letter? Because it actually denotes the same character. The same character, and these are the two different traits. Okay. So I don't know whether it is clear to you or not. So I will just move ahead with one or two more examples so that this becomes very clear. So once you know how to write the genotype for a particular trait, it becomes very easy for you to do the to show the crossing, the genotypic depiction of the crossing very easy. Okay. So now let us take another character. Because usually what happens, the student will just learn T and all that, and then it becomes very difficult for them if the question is slightly twisted and another character is given, right? So I am going to take another example. Let us take the example of the color of flower. Okay, so when I am talking about the color of the flower, now we know that we have two colors, purple and white. Okay, we have two colors, purple and white. Purple is a dominant trait and white is a recessive trait. Okay, right? Purple is a dominant trait and white is a recessive trait. Now how are we going to depict purple and white genotypically? So first, what will be our first step? Our first step will be to decide which alphabet are we going to use to depict this particular character. Okay, to depict the allele for this character. So I told you that we are going to use the first letter of the dominant trait. Okay, so the first letter of the dominant trait over here is what? P, that is purple. So I have decided that I am going to use the letter P to write down the alleles of this particular character. Okay, now let us talk about the purple. So since it is a dominant trait, so I am going to depict the genotype of this trait by writing capital P, capital P. Okay, because there are always two alleles responsible for this trait, and since it's a dominant allele, I am going to use the capital letter, okay, or the uppercase. So capital P and capital P. Then how are we going to depict this white? Yes, the lowercase of this particular, that is small p and small p. Okay, so this tells us that we are just talking about the same character, that is the color of the flower, which we decided that we are going to depict the alleles with this letter P. So the capital P shows that it's a dominant trait, and small p shows that it's a recessive trait. Is it clear? So the dominant allele is represented by capital P, the recessive allele is represented by small p. And capital P, capital, when I am writing two alleles together, so it's a genotype of purple, and this is the genotype of white. Is it clear? And can you tell me which purple, what would be the genotype of purple over here? We would call it as pure purple. Why? Because both the alleles over here are same. They are same. Okay. So when both the alleles depicting a particular character are same, we call it as a pure character. Okay. So now let's move ahead and take another example. Similarly, let us just take the color of seed. Okay, so we have two colors. One is yellow and other is green. Okay, and yellow is dominant, green is recessive. Right. Now, so how? So what will be the first step? Which letter are we going to use? Which alphabet to depict the gene or the allele for this character? So we are going to take the first letter of the dominant trait. What is it? Yellow, right? So we are going to use this letter, that is yellow, Y to depict the alleles or the genotype. Okay, the allele is Y, is represented by Y. So to show the to show the dominant trait, we are going to use capital letter, that is capital Y, right? And how are we going to represent green? It will be small y. So we will not use G. Okay, so don't get confused. Don't write G over here, because G means it's going to be another letter. So for a particular character, there should be only one alphabet. I hope it is very clear to everyone. Okay, so I'm just going to write down the genotype of, show you the crossing. Okay, the monohybrid cross. Fine. So first we will write P1 generation. How do we represent the tall? Capital T, and how do we represent short? Small t. Okay, right. So this can be taken, one of the plant can be taken as male, one can be taken as female, one can be taken as male. Okay. Now these are the alleles. Okay, so these alleles are going to combine, or it will go to the next generation. So how are these alleles going to the next generation? How will these alleles go to the next generation? So these alleles would be carried to the next generation by gametes, right? So we have to make the gametes. So our next step will be to make the gametes. So since gametes are always haploid, so only one allele for every trait will enter into the gamete. Always remember that. Okay. So we are going to write down the gametes. Okay. So one. So this is a female. So there are two gametes. Since there are two alleles. So one allele. And similarly for male also, you can say one allele and one allele. Okay, so these are the gametes. Now, during fertilization, the gametes are going to fuse, and again in the zygote, two alleles will enter. Okay, so we are showing gametes with only one allele because gametes are haploid. Okay, so now how do we show this crossing? Though there are several ways by which you can show the crossing, but I always prefer the Punnett square, the checkerboard method. I believe that things are. I think that it's very clear to you. I mean, अब देखिए ये हम इस तरह से बना रहे हैं. अच्छा, I am used to teaching in English, so हम फिर भी हिंदी में भी आपको चीजें समझा सकते हैं. ठीक है, तो यहाँ पे आप देखेंगे कि ये चेकरबोर्ड मेथड है जिसमें मैंने एक जो है स्क्वायर बनाया और इस तरह से दो कॉलम्स बना लिए और एक जो यहाँ पे मेल फीमेल, एक तरफ हम जो है फीमेल गैमीट्स लिखेंगे और एक तरफ आप चाहे तो मेल गैमीट्स लिख सकते हैं. ठीक है, अब देयर आर टू गैमीट्स, तो कैपिटल टी एंड कैपिटल टी है, और यहाँ पर मेल में भी दो गैमीट्स हैं, आप स्मॉल टी और स्मॉल टी. देखिए आप, आई थिंक इट्स वेरी क्लियर. ठीक है, अगर नहीं है तो मैं फिर से इसको प्रॉपर्ली लिख देता हूँ, स्मॉल टी. ठीक है, अब देखिए क्रॉसिंग हो रहा है, तो ये एक जाइगोट है और इसमें एक कैपिटल टी और एक स्मॉल टी है ना, एक मेल और एक फीमेल है, दोनों के एलील्स. ऐसे ही यहां पर भी हो गया, एक मेल और एक फीमेल एलील्स जो है वो गैमीट्स फ्यूज हो रहे हैं, है ना. तो आप देखिए, और ये हम चारों क्या दिखा रहे हैं, व्हाट एम आई गोइंग टू शो, आई एम शोइंग F1 जनरेशन, है ना, फर्स्ट फीलियर जनरेशन. एंड वी नो हाउ दे, व्हाट टाइप ऑफ प्लांट्स दे आर tall plants. Okay, तो अब यहाँ पर आप देखेंगे कि when you look at their genotype, so what type of tall they are? They are hybrid tall. What type of tall they are? Hybrid tall. Okay, so they are hybrid tall plants, right? And with this we come to the law of dominance, that whenever there is a heterozygous trait, in a heterozygous trait or in a heterozygous condition, for a particular character, one of the allele expresses itself and is known as a dominant allele, and the other one is known as a recessive allele. So a recessive allele never expresses itself in the presence of a dominant allele or in the presence of another allele for the same character. I hope it is very clear to everyone, the law of dominance. Okay, so I'll just continue this video, and I'll be talking about the law of segregation in our next video. Till then, take care.